Hi everybody, we're going to talk about membrane transport from the Chapter 3 information covering the cell. This information comes from page 35 in your course pack. Since this topic is called membrane transport, this is a great time for us to go back and review the plasma membrane structure. That's why it's called membrane transport, because this transport is influenced by the structure of this plasma membrane. So we oftentimes call it plasma membrane. You can also call it the cell membrane. And it does dictate what can travel through this external barrier to the cell. So you may recall that the plasma membrane is composed of two layers of phospholipids. Those phospholipids have polar heads, which you can see out here on the outside. And there are also nonpolar tails here oriented toward the inside. There are in fact two layers, and this bilayer creates a mobile or a somewhat fluid environment to the outside of the cell. These other fixtures you see here, such as proteins, are not solidified in place, you might say. They're actually able to move about as necessary through the plasma membrane structure. These proteins, by the way, are also critical for allowing us to transport items through the cell that may disagree, you might say, with those nonpolar tails, or may be too large to travel through those phospholipid bilayers. All right, so I just wanted to give you an intro there to the membrane transport by reviewing plasma membrane structure. And you may recall that plasma membranes are in fact selectively permeable or semi-permeable, and that means that they can select what travels through to the interior of the cell. In order to understand today's conversation, I need to discuss with you two terms, solute and solvent. The solute is the item in a solution that is dissolved by the solvent and is typically a lesser amount. So in the image shown here, this may be sugar being dissolved in water. The sugar is the lesser amount, it's being dissolved, therefore it is the solute. The solvent is typically the item doing the dissolving, it is the greater amount. And in our bodies, it may in fact be blood or intracellular fluids or even interstitial fluids. The solvent's not always a fluid, but it may oftentimes be. And of course, it's the greater amount doing the dissolving. So in this image, it's the water that's dissolving the sugar. The water behaves as a solvent. So let's talk about your bloodstream for just a moment. In your bloodstream, carbon dioxide may be dissolved by your blood plasma. The carbon dioxide is the lesser amount. It's also the portion here being dissolved. So in this example, what is the carbon dioxide? It's the solute. The liquid in the blood plasma happens to behave as your solvent. So there's more details to that than we're actually discussing here. That is something you'll hear more about in Bio 211. Let me give you one final example before we leave this slide. If you make sweet tea at home, sweet tea includes water, a tea bag, and some sugar. So let's say that you've boiled the tea, you've got the tea already made, and now you're ready to sweeten the tea. You may select a cup of sugar to place into your tea and stir until the sugar becomes dissolved by the tea. So in this example, what is the tea? The solution is actually a solvent made up of the tea, that's the liquid portion, and a solute, which was the sugar that was doing the dissolving. So in this PowerPoint slide, we'll talk about solute concentrations, and that's the proportion of the solute dissolved in the solvent. Okay, so I just want to mention this word because we'll talk about it. A solute concentration is the amount or proportion of the solute dissolved by the solvent. So if it's a really low number like 1 or 3%, that's not a whole lot of solute dissolved in that solution. So here you can see a solute and a solvent combine to create a solution. So we assume the solution includes both the solute and the solvent. So our goal today is to talk about passive and active transport. These are two ways for molecules or items to move across a plasma membrane to its interior. Sometimes the movement is what we call passive. Other times it requires active transport. So the major difference here between passive and active transport has to do with the use of energy. If no energy is required for the movement of molecules, from high to low concentration, we consider that to be a type of passive transport. By contrast, you'll notice active transport requires ATP, and sometimes those molecules are moved from low to high concentration. With passive transport, if we're distributing molecules along their concentration gradient, then we can reach equilibrium, meaning those molecules are evenly distributed on either side of that plasma membrane. However, with active transport, equilibrium will not be reached because we're moving those molecules against their concentration gradient from low to high concentration. So here's some examples we'll work through. Passive transport we'll talk through diffusion, osmosis, and facilitated diffusion, and we'll also talk about solute pumping, endo and exocytosis as examples of active transport. Now, there is a bit of room on your worksheet, bottom of page 35, if you would like to take a moment and jot this information down. The whole goal of this PowerPoint is to talk about all of this information in more detail. So here you can see another summary slide. Diffusion simply occurs by molecules moving from an area of high concentration to an area of low concentration across the plasma membrane. The goal here is to evenly distribute these molecules on either side of the membrane. You'll notice with facilitated diffusion, it's still a type of passive transport, but we are now using a protein channel, this integral protein right here, to help move those molecules across the membrane. So the goal is still the same, move those molecules from high to low concentration. By contrast, active transport uses ATP, whereas passive transport does not require ATP. Also, we move our molecules from low to high concentration in this example here, which is solute pumping, and ATP is required to move those molecules against their concentration gradient. So we're now going to turn over to page 36, and let's talk here about 3 types of passive transport. Let's start with simple diffusion, and then we'll walk through osmosis, and then facilitated diffusion. So simple diffusion is a type of passive transport. That means no ATP is required, and molecules spread from areas of high concentration to areas of low concentration. You just watched an animation. Those molecules did spread from an area of high concentration down here on the lower side to areas of low concentration at the other portions of this picture. Diffusion does not require a membrane for passive transport to occur. Diffusion can simply occur through the air, just as is done with, say, perfume that floats through the air at a party. So, in our bodies, however, diffusion does occur using a membrane many times, and so these molecules will travel across the plasma membrane without the aid of any ATP. They simply move with their own kinetic energy, bouncing off of each other's molecules and across the membrane. Gases often diffuse. In fact, carbon dioxide and oxygen diffuse across our membranes in our lungs, and then they transport throughout the body and diffuse again into our cells and tissues. That is the case of oxygen. So gases are a great example of how we see movement from high concentration to areas of low concentration. Now, in order for a molecule to diffuse across a membrane, that plasma membrane must allow for its diffusion. In other words, if the plasma membrane does not like the substance, that molecule cannot diffuse across the membrane. So if you think back, there are certain items that the plasma membrane does not like. Those phospholipid tails do not like water molecules. All right, so if you have a water-based item, it will not move easily across the plasma membrane. We'll have to find another route for that water to move. So some examples of substances that move by diffusion are small substances, those that do not break up the integrity of that plasma membrane. A second item that moves easily by diffusion is lipids. You say, well, why lipids? Well, the phospholipid bilayer has tails that are water-fearing, but they are lipid-loving. So the tails determine what can pass through the plasma membrane. And if you've got a lipid, of course the lipid can travel through with no problem. So on your worksheet, about halfway down page 36, make sure you jot down the two examples we just discussed. Water is not allowed across, but instead small items like carbon dioxide and oxygen can travel by diffusion, as well as lipids. So let's talk about what affects diffusion rates. In your body, temperature and viscosity play two major roles. If you have a cooler body temperature, diffusion rates will slow. This is something you should think about in terms of oxygen gas moving through your body. Higher temperatures speed up diffusion, so having a fever will cause the gases to move more quickly, and in fact our breathing rate has to speed in order to supply enough oxygen to the cells and tissues using that oxygen. The other thing that influences diffusion rates in our body is viscosity. That is known as the thickness of the fluid. Well, the one fluid that plays a major role here is blood. Blood is approximately 55% water. If you're dehydrated, that will cause the water to, of course, leave your blood, and the blood will become thicker. So we would say that a thicker fluid is more viscous. So if you're properly hydrated or perhaps even overhydrated or have anemia, then you may have thinner fluid or less viscous blood, and then diffusion can speed when you have a thinner fluid. If you have a thicker fluid, thinking here like, not maple syrup for your bloodstream, but if that gives you a mental idea, then that may be what you want to use. But the more viscous the fluid, the slower diffusion can occur. So if you're dehydrated, then we see that gases, for instance, move less quickly through our body. All right, so I'll leave you to finish the three main points you've learned so far about diffusion on the bottom of page 36. Let's now travel on to page 37 and talk about osmosis. This is still a type of passive transport. It is a special type of diffusion, however, because it is water movement across the membrane. So you recall that plasma membranes are semi-permeable or selectively permeable. That means that they can select what travels across the membrane. Sometimes certain solutes are not allowed to travel across the plasma membrane to equalize concentration. And when solutes can't travel to equalize their own concentration, water will sometimes travel across the membrane to equalize the concentrations instead. So let me give you an example here on the bottom of the page. What I'm drawing right now is a plasma membrane. Okay, this is outside the cell, and this portion here is inside the cell. Let's say that we have, a molecule. We're just going to call it this X here. Right now I have an equal number on either side of my membrane, so there's no need for water to travel in any direction to equalize solute concentrations. Now, this solute cannot travel across the membrane with ease. So if I start accumulating that molecule on the inside of my membrane, I now have a higher concentration of it. If this was simple diffusion, and if this particular molecule could travel through the plasma membrane, it would just simply travel from an area of high concentration to an area of low concentration. But unfortunately, that's not the case. This particular molecule cannot travel through this plasma membrane. So instead, in order to equalize solute concentration, water travels to where you have the higher concentration of this particular molecule. So water will actually enter this cell and travel to that area of higher concentration. That's what we mean when we say that the solutes can't travel across the membrane, but water will, and water will travel to where you have the highest concentration of this molecule. The whole goal here is to dilute down that higher concentration, wherever it is. The higher concentration may be inside your cell, or the higher concentration may be outside your cell. So, thinking completely different here. Let me give you just an example at home. If you've made your sweet tea and you didn't measure the sugar and you just dumped a bunch of sugar into your tea and you stirred it and you tasted it and you thought, wow, that's really insanely sweet. What if you had another pitcher of unsweetened tea ready to go? Could you fix the problem? Sure, right? You can pour the unsweetened tea into your insanely sweet tea mix and dilute down the sugar. So once you've mixed the sugar into the tea, you cannot remove the sugar, right? So it's in there, it's in the solution. But if you had some more unsweetened tea, you can dilute down the high concentration of that sugar by adding some more water. That's exactly what osmosis is. We can't do anything about the solutes. They can't move, or they have difficult time moving across the membrane, but we can move water to dilute down their concentration. So water will move from areas of low solute concentration to areas of high solute concentration. Don't let that confuse you. Water still moves from its own high concentration to its own areas of low concentration. Okay, so we're moving from low solute to high solute concentration, but we're moving from high water to low water concentration. Okay, that's just something to think about. We actually discovered aquaporins in the scientific community about 20 years ago, and we realized then that, oh, this is how we get water molecules across the membrane. They don't have to battle with those hydrophobic tails in the middle of the phospholipid bilayer. They can simply travel through a protein channel. You'll notice here it is an integral protein, and it's literally called an aquaporin, a water pore or a water channel. Now we understand how water moves from those areas of low solute concentration to those areas of high solute concentration. It is through these water channels called aquaporins. And in this particular animation, you can see that water molecules traveled across the membrane, but the solutes cannot. So the solutes are trapped on one side of the membrane. The water molecules, however, can travel freely. The one thing that's wrong with this animation, there are no aquaporins shown drawn into this particular plasma membrane. So that should have been there for us to facilitate the movement of those water molecules. This animation shows water traveling from an area where there's not very many solutes, in fact there's none on the left side, to an area on the right side where you do have a number of sugar molecules. For whatever reason, this particular membrane does not allow sugar molecules to travel from right to left, so in order to dilute down the sugar, the water molecules travel to the area where you have the highest salt or solute concentration, and that happens to be the right side of this picture. So this picture just shows how water moves toward the higher solute concentration. Water also moves toward the lower water percentage. Okay, so that is what we're thinking about here, is water moves from high water percentage to low water percentage in solution, but we're moving from low to high solute concentration. So when we talk about the issue here of moving something from high to low concentration as a type of passive transport, it is specifically the water that moves from high to low concentration. Let me walk you through the three examples here on page 37, and we'll start with the easy one. This is an isotonic solution. If we were to pluck a few red blood cells and place it in an isotonic solution, we would see that there's no change. The prefix iso here means same, and tonic means concentration. So let's just pick a number. Let's say that we have 3% solutes inside your cell, and let's say we have 3% solutes in the solution. Same, right? So the movement of water here is not going to cause this cell to shrink, or it's not going to cause it to bloat either. So the movement of water here, we say, causes no net gain or loss of fluids inside the cell. So an isotonic solution is one in which we see no net change in water movement. The cell maintains a relatively stable size. So if you could think of this as the cell's happy place, that's where it would like to be. It would like to be in equilibrium with its environment, and that's what an isotonic solution provides. So let's talk about hypotonic solutions now. Hypo as a prefix means under or below. Tonic, of course, we said means concentration. And let me draw your attention to a phrase in the course pack that says water always follows salt. I'm going to put that over here. Okay, so let's take our cell. Let's once again give it a 3% solute concentration. And let's say that we have it in a solution, but this time the solution is less concentrated at around 1% solute concentration. You'll notice my language there. I said that solution is less concentrated. Less sounds like under or below. So this particular solution is less concentrated. It's not at equilibrium with your cell. We know that water always follows salts, and the higher concentration of salt is found inside your cell. So we know that water travels toward the higher salt concentration. That means that water will enter this cell. As a result, this cell will bloat, and it takes in water until it reaches equilibrium with its environment. Well, that may never happen because this cell may eventually rupture, and that's a term that we call lysis. So hypotonic solutions end up having cells bloat in size and may eventually rupture. If you have trouble remembering this, remember that hypo has an O, bloat has an O. Okay, so that may help you remember that cells in hypotonic solutions will bloat. I've also had students share this in the past. Hypo sounds like hippo, and of course hippos are big, so hypotonic solutions will make your cells increase in size. So there are two reasons that water will enter this cell when it's placed into a hypotonic solution. One is simply because water follows salt, and second, it's because we are trying to reach equilibrium. That cell is trying to become as dilute as its surrounding solution, a 1% solution. Now, in a hospital environment, a dehydrated person may benefit from a hypotonic drip. The caution, of course, is that you don't let it run indefinitely without monitoring because their cells may ultimately lyse or rupture. So hypotonic drips can be great for someone who's dehydrated. Let's now talk about hypertonic solutions. We know that the prefix hyper here means above, beyond, or higher than. Let's take our same cell with a 3% solute concentration. And let's take a solution this time that's double that. Let's say it's 6% solute concentration. We have to keep in mind our general rule that water follows salt. All right, so this time the cell has not as many solutes as its surrounding solution. So we know that water follows salts, and water will travel to the higher salt or solute concentration. That means that we see water flowing from the cell into the environment. In this case, when water leaves the cell in an attempt to reach equilibrium, we see that cell shrivel. And the fancy word for shrivel is to crenate. Crenation. So don't confuse that with cremate, two totally different things, right? So cells crenate or shrivel when they're placed into hypertonic solutions. Now, if you keep this straight using the E's, that will help you out here as well. So let's take a person with heart failure, for instance. A person with chronic heart failure or heart fatigue is going to take on fluids and not be able to remove them, say, from the heart and lung areas. They may be given a pill that helps to reduce the fluids in their body, and if that doesn't work, they may end up in the hospital with a hypertonic drip. What does the hypertonic drip do to their cells? It pulls water out, so that may increase urination. So hypertonic solutions will actually dehydrate your cells, and that's the reason we're told not to drink salt water, because salt water will pull fluids out of your cells. In certain medical situations, that may be what we want to do, but in the general situation where someone is healthy, we would not want to take in hypertonic solutions. All right, so on your worksheet there are two reasons water leaves the cell when placed in a hypertonic environment. Well, one is because water follows salt, and there's more salt in the surrounding solution. The second reason is because we're trying to reach equilibrium. Water leaves the cell in an attempt to concentrate the solutes and make that cell as concentrated as the surrounding solution. So just a summary here, cells placed in hypertonic solutions, what will happen to them? Water exits, The cell crenates or shrivels. So what about a hypotonic solution? It has less solutes. This time water enters the cell may bloat and even rupture, a term we call lysis. The happy place for the cell is one with the same concentration of solutes. That's an isotonic solution. This picture shows red blood cells in an isotonic solution. On the left there you can see they're nice disc-shaped cells. And in an isotonic solution, the concentration is what? It is the same as the concentration of solutes inside your blood cell. Notice, however, here we see cells that have bloated. They've been placed into a hypotonic solution. Now, are hypotonic solutions more concentrated than the cell or less concentrated than the cell? They're less concentrated, and so water enters the cell because it contains more solutes. These are crenated cells or shriveled cells. What type of solution must they be in? One that's more concentrated or less concentrated? This hypertonic solution has more concentrated solutes than the cell itself, so water leaves the cell and causes the cell to shrivel. That's why they have this spiky appearance. That's proteins inside the cell giving that cell its spiky appearance. So, looking at this picture, back before refrigeration, people used to add a lot of salt to their meat, let it sit like that for a while, and created beef jerky. So, what is happening to the tissue when you put a bunch of salt on that meat tissue? Well, the fluids are pulled out to the area of the higher salt concentration, and it creates a tough piece of meat, obviously, but it also preserves that meat. So the fluids are pulled out, and that's what we're doing with a hypertonic setup here. All right, so in your course pack, I'm going to leave you to work on page 38 on your own and 39 and 40 on your own, and I'm going to take you over to page 41. All right, so let's talk about this type of passive transport, facilitated diffusion. Again, no ATP is required because it is passive, and we do move molecules with the concentration gradient from areas of high concentration to areas of low concentration. That's what it means when we say we're moving with that gradient. Now, this time we're going to be using protein channels to transport larger molecules like glucose. So in this image, you can see that these molecules take advantage of a doorway. This is created by a protein channel here to move molecules from area of high concentration to lower concentration. This makes facilitated diffusion different from regular simple diffusion because, one, we are moving larger molecules from high to low concentration gradient, and two, we are using a protein channel to conduct those molecules from areas of high concentration to areas of low concentration. When we get later on in this course, we'll talk about protein channels like calcium channels and sodium channels, and they are doing facilitated diffusion and moving molecules from areas of high concentration to low concentration. So we'll come back and revisit this thought a little later on. So we are talking about passive transport, and that facilitated diffusion conversation concludes passive transport. We are now going to move into active transport, which uses integral proteins sometimes, but those integral proteins behave as pumps, like you see here, and not as channels. So with active transport, we're going to be dealing with three examples: solute pumping, endocytosis, and exocytosis. All of this information on page 41. So solute pumping does exactly what it implies. We are pumping solutes against their concentration gradient from areas of high concentration to areas of low concentration. In order to move them against their gradient, we have to use this pump. And to power that pump, we have to pay it off using ATP. So you can see in this image, we moved sodiums to the outside of the cell, potassiums to the inside of the cell, and we do this against their gradients. So again, this pump that you're watching uses ATP to power the movement of these molecules against their normal flow. You're never going to reach equilibrium this way. In fact, that's not the goal of active transport. In this image you're watching, this happens on our muscles and nerves, and we don't want to create equilibrium. We want to create a polarized environment. And that's something we'll talk about in a later chapter. So let's talk about bulk membrane transport. This type of transport allows us to move vesicles and vacuoles across the cell membrane. One thing you'll notice here is the prefix endo versus exo is a great clue. Endo means we're moving those vesicles or vacuoles into the cell. What do you think exocytosis means? Moving those vesicles or vacuoles out. Now, there are two types of endocytosis. We have phagocytosis and penocytosis. Technically, the phago prefix means to eat, and the penocytosis means to drink. But more than that, phagocytosis means we're taking in large vacuoles, and penocytosis means we're taking in smaller vesicles. So it's the size of the sac that's being imported here that makes the difference between those two types. You're watching a cell engulf a sac. We could just say it's a vacuole. This is a form of endocytosis. We could probably say this is phagocytosis. Endocytosis may also form from invaginations of the plasma membrane, like you see here. The plasma membrane pinches off and forms a vesicle or vacuole that can then be moved about inside the cell. So this would be a way to import bulk items at one time. We actually get rid of bacteria by engulfing bacterial cells through endocytosis. You can see that process occurring here at the left of this picture. And the bacteria is degraded by lysosomes and their enzymes. The particles that are spit out on the right-hand side of this picture is a form of exocytosis. So we come full circle by bringing molecules or items in and then expelling them out through exocytosis. This is a great picture of an anthrax rod-shaped bacterium that's been engulfed or eaten by a white blood cell. The white blood cell is the yellow cell in this picture, and the bacterial rods are actually red in this picture. So you can see that the cell is doing all it can to transform its shape to engulf that bacterial cell. So great example of endocytosis. This specifically would be phagocytosis. This process here is, again, endocytosis. We're taking molecules in. They are formed from an invagination of that plasma membrane. That means that the plasma membrane forms a pit. It forms a circle around those molecules or vesicles, and then we create a sac for them to move into the interior of the cell. Exocytosis is the opposite process. It is the way we move large items or a bulk set of items to the exterior of the cell. And then in summary, we are looking at passive versus active transport. We have now wrapped up this information, and once again I'll let you take a look at this comparison chart and go back and jot that information down if you need to on a previous page. This comes from the bottom of page 35.